then the lights come on.
Okay, the lights were...
Well, I guess we're going to start right away. Thank you for joining us for this last session of the day. We really appreciate it. We're going to be talking today about tissue therapeutics and the promise of rationally designed tissues and to replace lost function. It's a topic that's close to my heart. I was in the tissue engineering field for a decade and helped take several tissue engineered organs to approval for a phase 1 clinical trial. So I'm here with a panel of experts today, key opinion leaders in the space, and they're all representing key companies in tissue therapeutics. So I'm really excited to be sitting here, and we're gonna kick off the discussion with first introductions. They're gonna talk a little bit about their company, and then we'll start in with the questions and answers. And hopefully we'll have time at the end, just a few minutes, for the audience to be able to ask questions as well. Okay, Petter?
Yes, hello everyone. It's a pleasure to be here. My name is Petter Björquist. I'm the CEO of a Swedish company called VERIGRAFT. So I have a long career behind me with regenerative medicine. I've worked for more than 10 years with cell therapies in other settings, but the last 10 years I have worked with something even more exciting, being tissue therapies. So my company, we are replacing failing cardiovascular tissue, and we also do personalization when we change the identity of a graft.
Thank you.
Great. Also a pleasure to be here, and great to see many familiar faces, although I know we're in between you and drinks, so we'll try to keep it exciting and engaging. So again, I'm Tamer, Mohamed, founder and CEO of Aspect Biosystems. Our company is based in Vancouver, Canada, and we're focused on creating implantable cellular therapies, mainly focused on metabolic and endocrine indications. We have a major partnership with Novo Nordisk, where we're advancing different programs in diabetes and obesity, and we also have our own internal pipeline that we're advancing in the rare endocrine and metabolic space. We have over 120 people in the company, brilliant scientists and engineers and therapeutic developers. And this topic is, of course, near and dear to me, so looking forward to the discussion.
Jeff?
Hi, Jeff Ross, president of Miromatrix, which is a wholly owned subsidiary of United Therapeutics, where our mission is to bioengineer transplantable organs to eliminate the organ transplant waiting list. Our key programs, our first one is in a phase 1 clinical study. This is our external liver assist product, where we actually bioengineer a liver, house that outside the patient to treat acute liver failure. And then our other area of focus is a fully transplantable bioengineered kidney.
Thank you. My name is Julie Allickson. I am the CTO for Mayo Clinic's Center for Regenerative Biotherapeutics. We have sites across the US in Minnesota, Florida, and Arizona. We have about 30 clinical trials in cell therapy, gene therapy, and tissue engineered products. So excited to be here today and start the discussion. And as we see, there's been a lot of, I think, a fair number of advances in the tissue engineering, and recently with Humanicite getting their BLA. It's very exciting to see that move forward. So just like to ask everybody if they'd comment on design innovations. What's the recent advances in design and development? Where do you think it holds promise for creating more effective tissue therapies? Because we have been in the space for some time, and there has been, starting out with the bladder. That was the first tissue engineered organ that came from Wake Forest with Tony Atala. And it's been a long time since then, and I think it's been a tough road. So maybe share with me what you think as far as advances that we see in the field. Petter, did you want to start?
I can start. So there are so many interesting things going on, so it's hard to pick just one or two of them. But one very important question over the years, not only in our area, but also in cell therapies, has been the question whether you should go autologous or allogeneic. But there is now more and more sign that you can turn these things around, so you can turn things that are allogeneic into autologous and make an advantage of that. So I see a lot of advancements there. Of course, that has traditionally been connected to maybe more complicated manufacturing and higher cost of goods, etc. So But we clearly see now that we can afford more lean manufacturing. So that is something that I'm coming into now, that we can manufacture at lower cost of goods, easily scale up these tissue therapies, because that is, of course, very important for the field that we can do so. The second thing I think about design now is not really the most or the best expert in, so I hand that over to Tamer, because I also see that printing and bioprinting is coming more and more into this, and that can be a bridge between different technologies. So in the past we have had a technology that I also know that Jeff is into, being D-cell and V-cell technology, where we now can see that we can use printing to overcome some of the challenges in that field. But that maybe is a logic turnover printing and DC-RC to the next two panelists here.
Yeah, happy to share some perspectives before I pass it on to Jeff. There's of course varying schools of thought when it comes to tissue engineering. The school of thought or philosophy that we adopt, that aspect, is very much one focused on the function and making sure that we are identifying the mechanism inside of the body that we're looking to replace with our cellular-based therapeutic, and using the technology to bring out that cellular function. An example of that is our work in type 1 diabetes, where we're not looking to recreate an entire organ. We're trying to replace a organ function that is lost in that particular area, where we're looking to incorporate cells in the right environment to restore that glucose sensing and insulin release function. Of course, that approach works for that particular indication, and we look to apply our technology in a way to not just effectively deliver the cells, but to keep those cells happy and providing the appropriate environment for them to function over a long period of time. And so the design questions then start to focus on durability of response and implantation and retrievability, to essentially provide the best experience for the patient, particularly when we're competing against current standard of care, which is a non-cell-based approach and something as commonly used as insulin. So I think there's no, I would say, one way to sort of approach sort of the design philosophy. It really is kind of indication specific and technology specific, and the view that we take at Aspect is very much one focused on function over form, and really starting from the indication that we're looking to address and working backwards, seeing how we could potentially make an impact.
Tamer, before we go to Jeff, can I just ask, do you think that there's then advantages, because right now they have the cadaveric islet cell transplants. I started in that field back in 2000, and a lot of those patients in the beginning required 2 and 3 organs to be able to render them insulin independent. Do you think that there's an advantage with the bioprinting?
Yeah, I would say, I mean, what's great about like the field of diabetes in particular, and using cell therapy as an approach, is like there have been advancements that build on previous advancements, and the work in the cadaveric islet space is certainly pioneering. I'm Canadian, and so diabetes is a place that's very near and dear to me because of the discovery of insulin, and also through the Edmonton protocol that was pioneered in Alberta. And so this is an area that we pay very special attention to, and the cadaveric work that was done in the islet space kind of gave us all a reason to believe that this is possible, but of course isn't a solution for the masses, given the requirement for immune suppression and given the lack of scalability of those cells. And so with our technology, we think we stand a great shot at addressing these challenges in terms of, again, incorporating a stem cell-based approach with a technology that allows us to effectively deliver these cells, immune protect these cells, retrieve these cells if and when needed. And our goal is to get into the clinic and prove that that's the case over the coming years.
I think, to follow up on Tamer's thoughts and question there, I think you hit on a key point in tissue therapeutics, and that really is the function over the form. I think a lot of times when you get into this, we have a preconceived notion of what that tissue should look like, how it should look histologically, but at the end of the day, it's really the function that matters. I think that's something we struggle with a lot sometimes in tissue therapeutics, specifically when we think about identity and potency. Now, the identity is easily the cells that we're putting in there, but the potency, we don't necessarily have to always look at the potency of the individual cells. We're looking at the potency of the construct. But to get back to your question a little bit, Julie, on that front, I think huge advances in 3D printing, certainly the resolution and the scale of that is pretty impressive on how that continues to advance the field. I think, broadly, biomaterials continue to get better. Our understanding of biomaterials continue to get better in terms of what we have to do to couple that with a cell to get that functionality. So I think that's an important advancement that continues to push the field forward. I think something that's really transcendental that I think is going to play a bigger role in the future as well is some of the stuff that we're seeing in normothermic perfusion. This is on the whole organ side. But as we develop these advanced therapies, certainly the thin ones, they can be cryoed. But when we really talk about therapies that are going to have this next degree of functionality, these therapies, you can't freeze them. You really need to be able to transport them as you would a tissue. And up to this point, it's really been cold storage, and cold storage has had its own challenges. So I'd say the adoption of normothermic perfusion, I think, is opening up new models for us of thinking about how we transport our tissues, but also how to extend that shelf life to make this much more adoptable in the future.
Yeah, I mean, there's others even looking at what can you add to that perfusion. So I think that that's going to be an important point as well. So as we talk about cell selection, what criteria would you use when selecting cells for engineered tissues? I know we touched on it a little bit, and the impact of the functionality of the final product, and maybe you want to start, Jeff, with that.
Yeah, for these, when we are tackling some of these problems, I try to keep a really, really simple philosophy on this and try to go as simple as possible. For us, we start with primary cells. So all of the cells that we receive today, we use those through organs that weren't placed for transplant. And the whole notion around that, as we look at bioengineering livers, bioengineering kidneys, we know today there's 100,000 patients in the US who are waiting for an organ. About 70,000 of those patients will not receive their life-saving technology this year, or therapy, but they're okay with immunosuppression. So our whole notion is start with a therapy, take that variable off the plate, let's develop life-saving technology and therapy to be able to provide that need to the patient. Once we're there, then focus on that next step of whether that's stem cell based for an autologous approach or whether you have a universal donor. So our approach is really stepwise. Start with potentially what's the simplest, try to take variables off the table if that's appropriate to scale, and then continue to move up more advanced to ultimately get to that holy grail. We all want tissue that you don't need to be on immunosuppression, that's fully integrated to the body. I think that's the destination. Yeah. The thought is, as you develop these, what's the journey and what can enable you to get to that final destination?
I agree. That's the grand challenge. We'd all love to see that. So thank you. Tamer?
Yeah, I mean, we always, again, start with the particular indication and then ensure that we have a very good understanding of the cell's ability to address that indication. So in the case of diabetes, making sure that that cell is capable of addressing the primary mode of action, but it could also be in the application of primary adrenal insufficiency that we're also focusing on, and making sure that that cell is capable of sensing the appropriate signal and producing the appropriate hormone. So we always start with really understanding the cells, because they are the heart of the tissue therapeutic. Everything else is just the house that the cell needs to live in, but the cells need to deliver that therapeutic effect. And then as it relates to autologous versus allogeneic, what's so great about the space is there's different views, and the approach that we're taking at Aspect is one that's focused on allogeneic therapies, where we believe that could unlock sort of the opportunity for the masses, which is why we're developing immune protection technology to avoid immune suppression. So this is something we think very, very deeply about at Aspect, because again, like I said, there's in my view, there's no tissue therapeutics without the cells, and the closer we could get to this being and feeling more like cell therapy and less like a science project, I think there will be broad adoption.
Yeah, I agree, definitely. Thank you. Petter?
So we can hear from Tamer and Jeff that two important things for this discussion. One is that it's always the function that is the goal. We need to cure a disease to help a patient. The other thing is that if possible, as Tamer says, if possible we should reach hundreds of thousands of patients, and then we can decide to do an allogeneic because that is a very good way of achieving that goal. But we can also, when we choose the cell population in our tissue engineered graft, we can choose to be a little bit more innovative than we have been previously. And I'm thinking about sometimes leaving the logic route. When you have your maybe a scaffold and you need cells to grow on that scaffold, you think which cells are I'm aiming for, then I harvest those cells. We may need a stem cell and we grow them, we isolate the stem cells, and that is typically too time consuming and too costly. So there can be innovative ways, like what we did was to think that we may not even need a pure population of cells. So we thought, we are in the cardiovascular area, what can the blood do for us? So we tried to use the whole blood as the source of cells, not to touch a single pure cell, instead only handle cells. And that turned out to be a great strategy. So that is how we have solved sourcing of cells and at the same time reaching more patients and lower costs of manufacture.
Yeah, that's fascinating. So you don't have to do any type of selections with that whole blood?
Exactly.
Okay, I'm impressed. So the session we had before the last session, which was led by Josh Ludwig, they talked a lot about cost of goods and scalability, which is very important, and bringing in automation and so forth. Can you talk a little bit about it from your perspective, from the products that you're working on? What can we do? What's the thought process to be able to look at the scalability and the cost of goods, and how can we reduce the cost of goods? So maybe we start with Tamer.
Yeah, so I was saying that the cells are, in our case, the heart of the tissue therapeutics, but they're also very expensive. So, and so they are the primary driver of the cost of goods. And I think, yeah, going through that analysis is incredibly important, and it's different for every indication, and there are many other considerations like the durability of this product and what is that trying to displace in terms of current standard of care, and what's the value that you could provide over that period of time, and even sort of cost benefit that you could provide relative to the standard of care. But the elephant in kind of the room is these are going to be expensive therapies. So there's no way around that. And as much, like, I think there's some financial engineering that you could do, or modeling as they like to call it, like to try to, but I think it needs to make sense, and you also have to, like, it needs to make sense in terms of the initial embodiment, and you would hope that costs go down over time as economies of scale tend to suggest. But I think we're still at the stage of, let's just get this to work, and at least it makes sense as a first step, and like you could reduce costs over time. But if your therapy requires cells, particularly your stem cells, it's going to be incredibly expensive.
I agree. Is there anything that you can say about the bioprinters? Because 15 years ago, when we were looking at bioprinters, I mean, there was a time where everybody made their own bioprinters, believe it or not. And then all of a sudden, some bioprinters started coming out, and there's 10 different opportunities for you to select from. And at that time, I would say none of them were completely GMP. Like, they were all needed to be tweaked to be able to get them in the clean room. But as we're thinking about cost with bioprinters, is that something that someone purchases and puts in their facility? Let's say the product gets commercialized, or as you're going down to clinical trials, or are they made somewhere else? Because it takes a lot of experience. I mean, you really need engineers or biomedical engineers to be able to make these products. So do you have any thoughts on that?
So our company spun out of the University of British Columbia about 10 years ago, and it was based off of an underlying microfluidic-based bioprinting technology. So we started with a platform, and then we landed on the applications that we were going to focusing on, diabetes being one, and others in the rare endocrine and metabolic space. And so naturally, as a company, we have a strong sort of underlying sort of technology focus and manufacturing capability. We've been kind of investing in that, and we're big believers of vertical integration, like just bringing everything under one roof. So we have a 50,000 square foot facility in Vancouver where we have our machines producing this material and all of the pieces in between. So we're big believers of that, and we're building a centrally manufactured workflow where the tissues are manufactured, and then they will ultimately be sent to the hospital sites around the world. And so we're developing the appropriate technologies to allow us to do that, but doing it in a phased approach. We're not setting a very high bar in terms of banking initially. Initially, a lot of these therapies will be live shipped to the different sites that we're targeting. But we are building a centrally manufactured process, and we have like a really strong team of people and the infrastructure to support that.
That's great, thank you. Jeff, did you want to?
Yeah, I mean, it's definitely economy of scale. I mean, I think the challenge with tissue therapeutics is one of the barriers to entry is you can't do this at a scaled down version. Like to do it in a rodent model isn't really directional for you because A, it's the wrong scale. You're not looking at how you got vascular supply and other things associated with it. And there's too much risk in trying to scale that up. So to be serious about tissue therapeutics, you really need to do it at a clinical scale or close to a clinical therapeutic scale, which is expensive to do for all the points that were talked about. It requires a lot of biomaterial, but it also requires a lot of cells and cell culture media. I mean, I think the hope is that once this gets to a commercial scale, and we've already seen it, as you continue to scale up, your media is usually one of your most expensive things in this whole cost of goods. But as you scale up, you're able to look at cytokine reduction at massive levels that really starts to bring that cost down. So I think as you continue to scale, you do see the cost of goods of this dramatically drop just because of the amount of purchasing power that you have at the scale that you're at.
Yeah, thank you. Petter?
Yeah, so I think this is the key of our whole area about scaling and cost of goods and so on. And in the world I'm living, and I'm sure many of you in the audience are living in the same world. With tissue therapies, you need something with the mechanical features like a scaffold, and you need something for the function, very often being the cells. And both are very often complicated to scale. So you heard my story about how we solved the cell source using blood instead of a pure population of cells that you have to isolate and purify, etc., and culture. And when it comes to scaffold, it's also, I think, about thinking out of the box. So we started with the lowest hanging fruit being a human donated scaffold, and that is perfect, but it's always harder to scale a human donated raw material. So our next strategy was to use animal material. We suddenly understood that they are working almost as good as human material. So a bovine vascular scaffold is very similar to human vascular scaffold, so we went for that. And then we are coming into the last step, at least so far, that we have done, and that is to print the scaffold. And remember, we are not talking about bioprinting here, we are talking about printing. So printing proteins is much easier than printing cells, I think. So that is a lower hanging fruit. So suddenly we have went down in costs, and then together with some innovative bioreactor developments, which is always also very important. I think we have been able to see just fractions of cost and much higher level of scaling than only a few years
Did
ago.
you develop your own bioreactors, or is that something that you?
We have our own bioreactor.
Okay, that's awesome. What about the D-cell process? I know, Jeff, you and Petter are both involved with the D-cell recell process. Are there opportunities for that to be the cogs scaled down or automate that? I know that can be very manual, and it's a, you know, it's
yeah, I mean, as you look at the cost of goods, certainly the matrix is of relatively low cost because we're taking an organ, in this case a pig organ, that would have naturally been discarded, take that in-house, we're able to decellularize that. So that part of the process is relatively cost effective. I think with the D-cell recell process, and then we take human cells, seed that onto the scaffold, and bioreactors continue to culture that and allow that engraftment and growth within the matrix. I think that does offer a really nice advantage in terms of cost of goods because we are able to seed 1/10 of the total cells that we really need and utilize the scaffold in the bioreactor to actually bring us up to that cell density. Whereas if we had to grow those cells, it would be much more expensive to try to get 500 billion cells before you seed a scaffold versus maybe you're seeding a scaffold with 10 billion cells and then allowing those to culture over time. So I think there's some clear, obviously I'm biased, but I think there's some clear advantages to the D-cell recell that helps keep some of those cost of goods down.
Awesome. So I'm just going to throw a question out about regulatory and that it's challenging in the tissue space. You've got combination products and, Tamer with the bioprinter and looking at the cells with that, it can be challenging as well. I know I've gone through some of that with FDA. ARM is extremely probably helpful with all the connections and so forth, but can you anyone talk a little bit about the navigation of that and what might be helpful for the audience that's, thinking about tissue engineering and how they would move forward with the product? Anything that you would, any advice from any of you?
I mean, I can start to kick it off with the fact that we got the first bioengineered organ through the FDA from a regulatory standpoint. And I think my advice to everyone was just take one step at a time. I mean, with a combination product and with tissue therapeutics, you will be a combination product. CDRH in the US is going to do your biomaterials or your mechanical component of your scaffold, and then CBER is going to be the notified body for all of your cellular therapies. They're going to be the main one because at the end of the day, the mode of action is the cells. But I think you just break it down, and luckily with ARM and other things, the foundation has really been laid. If we look at medical devices, they've done a great job at commercializing biomaterials. So you know what you need to do from that standpoint and what you need from quality control, what you need for viral clearance, if you need that, sterility, 10993 testing from a biocompatibility standpoint. There's a really good recipe book on that side. I think all the great advancements that we see in cell therapy have really allowed and laid the foundation to get comfortable with how do you mass produce cells? What does that look like? What's your release criteria? I think the new combination that you need to work with on the agency is, well, what does that look like when you bring them together? And I kind of hit it on it in a little bit in the beginning. From a biomaterial standpoint, you need to worry about identity and potency, at least not in those terms. From straight cell therapy, yes, you need to show identity and potency prior to initiating that therapy. Well, when we start to combine those in a tissue therapeutics, what does that look like, right? For us, a lot of identity on the cells that we're putting into the process, but once they're in the process, we all know that there's multiple cells in these constructs, at least for us, and they interact with each other in different ways. So how do you start to define what potency looks like? Do you do that at the beginning of your process? Do you do that at the end of your process? And I think having that answer and understanding how you're going to tackle that's really important as you do move through that regulatory process, because that's going to be one of the key elements that you need to tackle.
I think it's important also to have those early discussions, whether it's European or MRH or FDA, and there's opportunities, I know, with FDA to be able to go in and discuss with a platform technology early on, calling it a CAT, to be able to get their feedback on some of the questions. So I think earlier the better is really important. So we'll take one question, then we'll have a final question for the panel. Is there any questions in the audience? Everybody's quiet. So I guess I would like to hear from everybody, where do you see the future of these tissue therapeutics? It's only a very small section of, like biologics in general. And so there's not a lot of experience with commercializing these products. So, you know, what do you see for the future? And I guess also, what do you see happening and what do you hope for the future? So, Petter, do you want to start?
Yes. We have talked about several very important things today. About the future, I would like to start to say that I think we are now at a time where we should not see tissue therapies as something extremely complicated any longer. I mean, we have always been seen as the complicated sibling in the family, sort of the cell therapy and gene therapy. They are complicated, but tissue therapies, they are far too complicated. So we can't commercialize them, and we can't do that. So my first message is that I really think that we should start to see tissue therapies as something important for medicine and something that we can and we will commercialize in a good way. I know that many tissue therapies are far more or far less complicated than some gene and cell therapies. So I think we should be more accepted when it comes to that. And having said that, I would like to take the opportunity. I'm not sure how many ARM members we have here. We typically, or ARM staff people, but we used to put a lot of good words here. I think it's time when we talk about this field as CGT, not see CGT as cell and gene therapy. It should be cell, gene, and tissue therapies, right?
I love it.
So it's time to accept us more as an equal member of this family. That is how I see the future. So having said that, my last word, I see the time is running, is that I also think what I see, that tissue therapies not only will be for orphan diseases or small number of patients. I strongly think that with strong technologies as Jeff's and Tamer's companies, and hopefully my own as well, are now able to meet large population of patients. So many patients, hundreds of thousands of patients in the future.
Thank you. Tim?
Yeah, the only thing I would add is at some level we're all on the same team, not just sort of tissue therapeutics, but broadly speaking, any cell-based approach. And so success is what everybody needs to sort of fuel the entire space. So really like the pragmatic and rational approaches that are being taken by both Petter and Jeff, and just getting to the clinic and showing efficacy and patient impact, I think is important, because the beauty with the different technologies that are being developed here is I don't think anybody would disagree that they have transformative potential. I think to Petter's point is how you could reduce the complexity to something that's simple and proven is what people have concerns with, and so getting to the clinic and getting to patient impact is what I think the future has in store, and I think a lot of the hard work the past several years is coming head on over the next several years, and I'm wishing everybody here success that is trying to do that.
Thank you.
I think to add on a little bit, I mean, the complexity, it does sound complex, but glad you said that, Tamer, and Petter, you brought that up, and in many ways, there's some things that are simpler. I talked a little bit about the potency. I mean, at the end of the day, I do think sometimes a potency assay is actually easier. And why that is, at least for us, if you're developing an organ, let's say we're looking at a kidney or we're looking at a liver, we can test the function of that organ in a true functional assay prior to ever releasing the facility. So instead of trying to guess what the potency and predictability of a cell is on its final destination, we can actually assemble the whole thing and say, does it do the functionality we're looking at? So in some ways, I find that more
That's a great point.
Easier, right?
Yeah.
Instead
Yes.
of the complex of trying to choose your potency. And if you get it wrong, you got to go back and correct that, because now you're off, even though your therapy is effective. I don't know if you want to add anything on that.
Yeah, I mean, I'm just agreeing.
Yeah,
I
that's
think
great.
it's such a great point. I think because we're using cells, like, I think you could come at it from a functional perspective. Like in the case of like the indications that we're going after, like the question of overdosing is something that could be on your mind, but as long as the cells are performing as they should, you can overdose with the production of the hormone, for example. And so that's something that you can't say is the case if you're going after a traditional small molecule, or where you have to make sure you're not going to overdose the patient.
Right. And then to answer your final question, I mean, I'm really excited about the future. I do think that these are the early stages of tissue therapeutics. I think we need some big wins. We're starting to see all these therapies start to get into the clinic, which is really exciting. But I think if you even take a step back and take a holistic approach, I mean, if you look at the advancements in cell and gene therapy, the future is bright there to solve many of the challenging diseases. I'm really hopeful on solid tumor and the efficacy that can come out of that. With all of that, people are going to be living longer. Some of the things that we're solving in tissue therapeutics, I don't believe can be solved with tissue and gene therapy. So with that, I think it's going to bring even that need for these advanced therapies to a greater and greater level. So I'm excited to start to see some of these wins get through the clinic, start to have that impact with the patient. But I think it's just going to accelerate the whole field, because there's a lot of need on the tissue therapeutic side once damage has been done that you can't replace any way else except actually replacing that tissue.
Yeah, no, I agree. I'm excited. All the things that you've brought to the table, the experience, and just the little tips that you've provided today. I know we're step by step going there, and hopefully someday we will be at the future of being able to bioprint or decell where they won't require immune suppression. But these are the steps that we need to take to be able to be there. So super excited, and I really appreciate everybody sticking around for the last session. So I'd like to thank our panel members and key opinion leaders in the field of tissue therapeutics, and thank you for joining us. That was great. Thank you.
Thank you.
Oh, nice job. Good.
Oh, thanks.
Thank you. Good to